Chapter 1, Cellular and Molecular Biology of AqueousHumourDynamics from Shield's textbook of Glaucoma, 6th edition, page number 4, Embryology of the Eye. The eye shows incredible diversity among the various phyla, from simple eye spots to compound eyes to complex structures with a single lens and photoreceptor arrays. The developmental biology of the vertebrate eye, from surface ectoderm, neural crest, and mesodermal mesenchyme, has been extensively investigated. An overall schematic of eye development is summarized in Figure 1.3. The tissue origin of the various ocular structures is summarized in Table 1.1. Ocular development from these three tissue sources involves complex specific cell growth and differentiation processes, which are not fully understood. These complex processes involve carefully timed expression of various growth factors and their receptors, other signaling molecules and their pathways, transcription factors, and structural components. In general, the genes that regulate development can be categorized into different functional classes as follows. A. Structural genes such as cytoskeletal components, which may be considered as housekeeping genes that carry out ubiquitous biochemical and structural functions. B. Regulatory genes such as transcription factors, that is, molecular switches that control mRNA production by other genes, and cell signaling molecules, which mainly determine specialized expression of genes, and C. Cell-specific genes encoding for specialized proteins of a particular cell type within an organ, such as the unique proteins expressed in the photoreceptors, abnormalities in expressions of the individual genes, or interaction among multiple genes caused by gene mutations or altered expression can lead to congenital defects and human disease. The following regulatory genes have been grouped into large families of transcription factors: homeobox genes, zinc finger genes, and helix loop helix genes. Homeobox genes encode for a 60 amino acid DNA binding element and specifically determine the target gene for a transcription factor. These genes are frequently involved in determining the regional identity of the embryo or individual fate and differentiation of cells. Examples of homeobox genes include the Parkes family and POU domain family. The zinc finger family of genes is thought to be the most abundant of the transcription factors. These genes share a common motif of a zinc atom binding to a group of histidine and cysteine amino acids and holding together a small loop of amino acids. Examples of this gene family include the retionic acid receptors, RAR, and retinoid X receptor, RAX, which direct the binding of retinoic acid. Mutations in these receptors have been associated with abnormal eye development. The helix loop helix family of genes is characterized by two helical DNA binding domains held together by a spatial domain or region called as leucine zippo. The role for these various structural, regulatory, and cell-specific genes in ocular development has been most extensively examined thus far in the retina, which is highly complex and only partially understood. Although not as extensively studied as retinal development, the anterior ocular segment, including the ciliary body and lens, also has important and complex roles in the development of the normal eye. The tissue origins of the ciliary epithelium, ciliary smooth muscle, and lens are listed in Table 1.1. The lens induces differentiation of ciliary epithelium at the edge of the optic cup, and the iris develops later from the edge of the optic cup. The ciliary muscle and stroma differentiate after the ciliary epithelium is formed. It is not clear when during gestation the ciliary epithelium becomes active to secrete aqueous humor, but it is assumed to start very early after formation. As the IOP increases, the eye grows. It is also believed that the increase in IOP provides the force to generate ciliary folds in the ciliary body and to change the shape of the cornea. Abnormalities in the development of the anterior chamber angle and anterior segment dysgenesis are exemplified in Axenfeld-Rieger syndrome. Thus far, genes that have been shown most frequently to cause anterior segment dysgenesis encode transcription factors that are important in early development. These transcription factors include PITX2, PITX2, PITX3, PITX3, PAX6, PAX6, FOX7, FOX7, FOXC2, FOXC2, and FOXC3, FOXC3. In transgenic mice, the cell signaling molecules, bone morphogenetic proteins, and related signaling molecules play an important role in normal development of the anterior segment. An approach to study embryology of ocular structures is using data obtained through bioinformatics, a discipline that integrates the study of genes, pathways, and function. Gene expression data, also shown as transcript or mRNA expression, may be gleaned in discrete ocular tissues and at various time points in development. Such a global overview of gene expression in this discrete ocular tissue enables us to hypothesize and to design studies to answer some fundamental cell biology questions about these ocular structures. By comparing and contrasting the gene expression profiles of these discrete ocular tissues at various stages of development and the impact of environmental exposures, we will understand the function of these eye structures at the cellular and molecular level. Figure 1.3, schematic of early eye development from the optic vesicle stage A, lens precoce stage B, and optic cup stage C. During the optic cup stage C, the neurogenesis of the retina proceeds in a highly regulated process with ganglion cells differentiating first, followed by the amacrine cells, bipolar cells, horizontal cell photoreceptors, and molar glial cells. Table 1.1, derivatives of embryonic tissues. Neuroectoderm. From neuroectoderm, following structures are developed: neurosensory retina, retinal pigment epithelium, pigmented ciliary epithelium, non-pigmented ciliary epithelium, pigmented iris epithelium, sphincter and dilator muscle of iris, optic nerve, axons, and glia vitreous. From cranial neural crest cells, following structures develop: corneal stroma and endothelium, sclera, see also mesoderm, trabecular meshwork, sheaths and tendons of extraocular muscles, connective tissues of iris, ciliary muscles, choroidal stroma, melanocytes, uveal and epithelial melanocytes, meningeal sheaths of the optic nerve, Schwann cells of ciliary nerves, ciliary ganglion, most orbital bones, cartilage and connective tissue of the orbit, muscular layer and connective tissue sheaths of all ocular and orbital vessels. From surface ectoderm, following structures develop: epithelium, glands, cilia of skin of lids, and caruncle, conjunctival epithelium, lens, lacrimal gland and drainage system, vitreous. From mesoderm, following structures develop: fibers of extraocular muscles, endothelial lining of all orbital and ocular blood vessels, temporal portion of sclera, vitreous. Table 1.2, Gene family, OTX2 homeobox. Its function is regulatory. Tissue expression is found in retinal pigment epithelium, optic nerve. Animal model, mouse knockout, lethal. Human disease not reported. Then second gene family is PAX2 homeobox, PAX2. Its function is regulatory. Tissue expression is early optic nerve, kidney defects. Animal model, mouse knockout eye, kidney. Human diseases, renal coloboma syndrome. Gene family, PAX6 homeobox, PAX6, PAX6 homeobox. Its function is regulatory. The tissue expressions are found in lens, retina, nose, brain. Animal model is mouse small eye, Drosophila eyeless. Human disease, aniridia, anophthalmia, Peters anomaly, brain nose defects, optic nerve hypoplasia, coloboma, microphthalmia. PETX2, bicoid homeobox. Its function is regulatory. Tissue expressions are brain, pituitary, ocular mesenchyme, cardiac mesenchyme, neural crest. Animal model studies are chicken, frog, mouse, situs inversus. Animal model, chicken, frog, mouse, situs inversus. Human disease, Axenfeld-Rieger syndrome. Gene family is POU, BRN3, RPF1. Its function is regulatory. Tissue expression is retinal ganglionic cells. Animal model, mouse knockout, ganglion cell hypoplasia. Human disease not reported. Gene family, thyroid receptor, TR. Function is regulatory. Tissue expression, oligodendrocytes. Animal model, mouse ganglionic cell degeneration. Human disease not reported. Gene family, XATH5, basic HLH. Function is regulatory. Table 1.2, selected genes involved in vertebrate eye development. Gene family, BMP4, TGF-beta, regulatory. Tissue expression, optic primordium. Animal model, mouse anterior segment dysgenesis, IOP, abnormal teeth. Human disease not reported. Then BMP7, TGF-beta, function is regulatory. Tissue expression, optic primordium, cornea, kidney, skeleton. mouse knockout, microphthalmia, mouse polydactyly, human disease not reported. BRN3B, POU domain, regulatory, tissue expression retinal ganglionic cells, animal model, mouse knockout, optic nerve, mouse hypoplasia, human disease not reported. Gene CHX10, homeobox, function transcription factor, tissue expression retina brain, animal model, mouse ocular retardation, human disease microphthalmia, cataracts, abnormal iris sclerocornea. Gene CRB1, function structural, tissue expression retina, animal model, Drosophila photoreceptor abnormalities, human disease, Leber congenital amaurosis, retinitis pigmentosa. So gene family CYPB1, it's regulatory. Animal model, mouse anterior segment dysgenesis, human disease congenital glaucoma. Gene gamma crystalline, beta gamma crystallines, structural, function is structural, tissue expression lens, animal model, mouse eye lens, obsolescence, ELO, cataract, human disease, Kopok cataract, congenital lamellar punctate and nuclear. Gene FOXC7, FKHL7 or FREAC3, Bicoid homeobox, its function is regulatory, tissue expression anterior segment of the eye, animal model, mouse hydrocephalus, skeletal and eye abnormalities, human disease Axenfeld-Rieger syndrome, anterior segment dysgenesis. Gene LMX1B, homeodomain, regulatory, tissue expression anterior segment of the eye, animal model, mouse microphthalmia, human disease Neil-Patella syndrome with COAG, that is chronic open angle glaucoma. Gene MAT3 basic HLH, MATH3, its function is regulatory. Then gene MI basic HLH, its function is regulatory, tissue expression retinal pigment epithelium, pigment cells, animal model, mice microphthalmia, human disease Waardenburg syndrome type 2, Tietz albinism deafness syndrome. Gene MYOC, 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 function is structural, tissue expression trabecular meshwork, ciliary body, iris muscle, animal model, fluid discharge in the dorsophilas, human disease juvenile glaucoma. Gene NR2E3, function is regulatory, tissue expression retina, animal model mouse retinal degeneration, human disease enhanced S-Cone syndrome, Goldman-Favre syndrome. Gene OCRL1, inositol phosphatase, its function is regulatory, tissue expression lens brain kidney function, animal model mouse knockout without Lowe syndrome phenotype, human disease Lowe syndrome. Gene OPTX2, bicoid, tissue expression retina, animal model mouse pituitary retinal and optic nerve hypoplasia, human disease anophthalmia. Gene OTX 1/2 homeobox, function is regulatory, tissue expression iris and ciliary epithelium, ocular surface, animal model mouse knockout brain seizures, mouse lacrimal gland missing, human disease not reported. TGF beta stands for transforming growth factor beta. IOP stands for intraocular pressure. COAG for chronic open angle glaucoma. HLH means helix, loop, helix. Thank you. Enough for today.